Today, I'll be talking about some of my work looking at how perception interacts with memory. And I'll be doing this through methods and lens of cognitive neuroscience. So the plan for today is I'll be talking first of all about how once we have knowledge, once we have memory, how does that influence the visual system? Next, I'll be thinking and talking about how once we have visual variation in the world, how is that bound together and how does that in turn influence the kind of memories that we form and how is it formed in the brain? Third, once we have new knowledge, I'm going to be looking at how that affects the nature of new learning. And then finally, I'll be thinking about really very naturalistic, dynamic visual episodes, the kind of thing that's happening to you right now if you're watching me. And I'll be talking about how we are able to, and looking at how we're able to extract information and put this information into memory. So first of all, memory influencing the visual system. So when I think about semantic knowledge and how our memory intersects with perception, I always come back to this classic diagram, this classic viewpoint of semantic memory. And whether you happen to subscribe to the more distributed view, or perhaps a distributed view with some kind of hub in the brain that brings together information from different sites, either way, as you can see from this diagram, it's pretty clear that distributed areas are involved. And the kind of areas that we believe and know to be involved in semantic memory are very much similar or overlapping with those areas we know to be crucial and involved in perception. But for me, one thing I found this really, why I found this really interesting is there's kind of a challenge here, not just understanding in where or what areas are connected to each other, but really how information is exchanged between them. How does the sharing of information lead to semantic memory and how is it used? And this is particularly an intriguing challenge because some of these areas in these perceptual cortices are fairly specialized, right? They have particular neural machinery. So for it to be used both in perceptual terms, but also interfacing with knowledge is really an intriguing idea and an intriguing challenge. So to look at this, I'm going to be looking at a property, a characteristic that really is a great way to look at how perception and our understanding of concepts, percepts and concepts comes together. That's real world size. You might also know this or think of this as canonical size. So as we go around our daily lives and as you look around objects and items around you, there are cues, visual cues that can tell you its size. Okay, it might be the depth, it might be its relationship to other items around you. And these visual cues can help you, as I said, determine size, but these aren't always sufficient. So for example, we may have an item that we just don't know the natural real world size of. Similarly, there are items in our environment that look really similar to each other, have a similar shape and so on, but are actually different sizes. And actually, as it happens, since I've kind of pursued this line of work, a side hobby of mine has been collecting images of items that look visually kind of identical or very similar, but actually are different sizes. And you can kind of see this in this image here. In fact, you maybe wouldn't be able to fully process the size until you actually look at the hand, right, or the glove to actually determine the size of each of these. And it's also the case that in our daily lives, we use our knowledge of size to understand and to make predictions about the world around us. Let's take a look now at this in this clip. So here our expectations are violated, right? And our expectations are based on our knowledge of size, how large a telephone typically is. And the humor, the absurdity that comes from this reflects the fact that we are making predictions based on our knowledge. So what I'll be doing now is I'm going to be talking about two studies, two separate studies, investigations to understand how real world size is represented in the brain. So what do we know about this? Well, we know that the real world size of objects, like man-made objects or artifacts, does tend to be accompanied by a larger response, a larger bulb response or fMRI response in areas of the brain that are involved in processing high-level vision, such as ventral temporal cortex. However, this is a little bit more trickier to separate out than you might think, because object size, at least for man-made objects, is often confounded. It's confounded with landmark potential and manipulability, namely that big objects have the potential typically to be landmarks, and small objects, in contrast, have the ability to manipulate it, okay, but not the other way around. And this is really, these are two important properties of the brain, not least for the ventral temporal cortex, which responds very heavily to places and scenes and landmarks, but also to the dorsal stream, which is involved in action processing, which in turn feeds back to the ventral stream in a number of different ways. So it's very difficult to draw conclusions about object size when we just have this kind of like innate confound here. However, animate items provide a really nice solution to this, because animate items, such as animals, are mobile, so they're not particularly good landmarks, and also they aren't really manipulated. I mean, maybe if you're a farmer and you're carrying a chicken around, but for most of us, we're not really frequently thinking about how to like manipulate a particular animal. So animals are great for this, but past work has found that animals or animate items don't produce the same kind of size-based fMRI response as man-made objects. But after seeing some of this past work, one thought that occurred to me is that some of the ways that these past studies might have been asking this question may not be actually getting at how the brain is representing this information. And this is where I'm going to turn and introduce you to a concept that's going to come back several times, for those of you who aren't familiar with it, of looking at multivoxel patterns. So a lot of the fMRI studies I'm going to be talking about will be detecting or trying to detect or identify multivoxel patterns in the brain. So what does this mean? Well, a typical more traditional or older approach to looking at fMRI data will look at a univariate signal, okay? Really like a peak, right? A peak response. And just like these kind of icebergs shown here, a peak response, often thresholded in some way, is very informative. It shows you, the location in this case of something, and also clearly reflects some kind of signal. But just like icebergs, there can be a lot more information kind of underneath the surface. And something that can look quite similar in pattern from its peak can actually have a very different formation, organization, once you look at kind of the full picture. And so multivoxel patterns in the brain are a way to try to get a bit more of a subtle fingerprint, neural signature representing things. So to go further, here's a diagram I find particularly useful. We can imagine that we have a particular brain area. Within that brain area, we've got a series of voxels, right, or 3D volumetric pixels. And in this particular case, we've got sort of 9 of them, right? These are particularly large voxels in this diagram, but we got about 9, and we can see here that each of these items, robin, parrot, chair, sofa, has a unique sort of pattern of activation in each of these. And a standard fMRI analysis will ask, is there a difference in activity between these voxels or these regions? The kind of techniques I'm going to be using, namely MVPA and also some related methods, look at the patterns and ask, can we distinguish these items based on the unique pattern here? And we use for this machine learning classifiers and also really similarity metrics. So for example, if we look at this diagram here, in this case, perhaps a classifier has been able to lock on to a reliable, robust signal here and is able to determine when you have these birds versus these objects. And if a classifier can do this using the activity from an area, as long as we control for other things, we can infer that the region itself has the information needed to separate them. Similarly, to the right here, we can ask about the structure and organization of information in a region by asking how similar these patterns are. So in this sort of toy example here, we can see that robin and parrot, those patterns are pretty similar to each other. And you can kind of see that a little bit here, right, in the diagram of the voxels. But they're quite different from chair and sofa, and vice versa. And so here we can see that the patterns for these birds look more similar in this region, and the patterns for the objects look more similar to each other. And so that can tell us a little bit about how this area is organized. So I'll be using these techniques throughout this presentation. So how have we gone about understanding real-world size? Well, in this first experiment, what we did is we modeled real-world size in the brain while accounting for factors that typically confound it. Okay, so we presented people with a series of images of animals which were selected in a very careful way. These animals were selected based on dimensions, which we also know to be important for the kind of areas we're interested in. Familiarity, so half the animals tended to be familiar to people, half were not. But also, size tends to be confounded or at least correlated with taxonomic category. In short, mammals tend to be bigger than birds, and birds tend to be bigger than insects. And that becomes a problem for us trying to understand real world size. So to get around this in this study, we actually selected items that kind of break that confound, break that continuation. So we actually deliberately chose that some insects that scarily are larger than birds, and some birds, ostrich for example, that are larger than some mammals. So that allowed us to get to size without hitting against this kind of confound that limits us. I should also say, although some of these animals were not familiar, just to make sure that in this study everyone at least understood the real world size, we did show them the day before wildlife videos of those animals sort of in action to give them the context so that they weren't coming in with no size information at all. Oh, as a sort of note as well, if you find yourself, I don't know, a little distracted during the presentation and you want a little game, you could count the number of times the shoebill, this creature here, appears in this presentation. It's a surprising high number. I thought it would be quite a lot. It's even more than I thought. So something to keep in mind. So let's jump into some results. So we show these people images of these animals, and these are different exemplars. I showed just particular views here, but we showed lots of different views and different forms, right? Different individuals, if you like, of each of these species. And so what we first did is we looked at pattern similarity, so how similar the patterns are for these different animals, and saw how that tracked real world size. And we did that, well, like I mentioned earlier, partialing out taxonomic category, familiarity, and also visual features. I don't have the time to go into the details of how we did that here, though as with all of this, happy to dive deep into that for the questions, but we had models that accounted for or at least quantified the visual features. So what kind of results are we going to see here? Well, we're looking at a correlation because we're looking at the relationship between how similar, the pattern similarity between items, let's say elephant and rhinoceros. Okay, they're both big animals. So we're going to look at how the brain represents, say, these weren't stimuli, but an elephant and a rhinoceros. And we're going to see, and if the brain indeed represents size, that although there's a very small difference in the size between an elephant and a rhinoceros, it should be very similar in the brain if size is being represented. And when we look at areas throughout the ventral stream, starting from the back of the occipital cortex through the temporal lobe, we see that surprisingly early areas, areas associated with early visual cortex, show this negative correlation, suggesting they represent real world size. We went on to do a similar analysis, but this time just to restrict the pairs that we were looking at to within taxonomic category. Basically, we made sure there weren't any pairs or similarities that cross the bird-insect boundary or the bird-mammal boundary and so on, just to make absolutely sure that taxonomy wasn't interfering. And yet again, we saw that the early visual cortex, early visual areas seem to represent real-world size. So how else can we look at the areas that might be responsible or might hold real-world size knowledge? Well, in a second experiment, we taught people real-world size, and we looked at the effect of teaching people this information on brain activity. And real-world size can be taught, right? I can show you this insect here, and you might not know exactly how large it is. And I could show you the real size in a few different ways. I could show you it in context, like this, so you get an idea. Or I could tell you it. I could verbally tell you it, and we can learn it declaratively. And I can say, this insect is about 4 inches long. Either way, you might be horrified, but in both cases, hopefully you've learned the size. So what we did is we introduced people to animals that they tended not to be very familiar with, the tapir here and the echidna, as well as some other items, tools, kind of like our control, or at least a different category of items here, the wood plane and the pump drill. And after a kind of a pre-learning scan where people were looking at individual different exemplars and different viewpoints of each of these items, we then introduced people to the size of these animals. So for example, here they might see the tapir and be told this animal is between 6 and 7 feet long when they're fully grown. And there was a sort of an intervention. They had to imagine the animal. They had to respond to a kind of a factual question about the animal to make sure they really learned it. And we also had for the tools information about how you actually operate, how you manually successfully operate the tool. I'll be focusing on the animal results, for the size in this talk. So how do we measure the size information? Well, I didn't mention this earlier, but as well as these new animals, the tapir and the echidna, and we assess familiarity as well to make sure they're unfamiliar for people. But as well as those, we presented animals that they knew. In particular, the raccoon, which is very similar size to the echidna, and a sheep, which tends to be approximately a similar size to a tapir. And so what we did is we were able to look at how similar the patterns in the person's brain were between the new animal and animals that they know, to look at a change. Now, at no point did we say something like the echidna is about the size of a raccoon. We never actually explicitly told them that. We just showed them raccoons and separately told them in feet and weight and so on, the information about each of the new animals. So we looked at early visual cortex and later ventral temporal cortex. So in this blue region here, early visual cortex, we found that after teaching people the size of the animals, that these new animals became less classifiable with animals of the same size. Put another way, an echidna became more confusable in early visual cortex with a raccoon. And a tapir became more confusable in early visual cortex with a sheep. And in terms of confusability, by the way, I don't mean that the person, I should say, was kind of confused in what they were seeing. I meant confusability of the patterns. So the patterns are just more similar to each other. That didn't happen in VT, later ventral temporal cortex. In fact, it went the other direction, probably because if you're learning more information about an animal, that's going to help you distinguish them, right, in other dimensions. So these other dimensions, like how typical a mammal it is or something like that, might be really important for other areas. But they became, after teaching, the size information became represented in early visual cortex. And actually this kind of approach to using classifiers, where we're interested not just in whether we can classify, whether we can distinguish, but what's confusable, actually motivates some ongoing work that I'm doing now with Dr. Julie Fees and Dr. Melissa Libertis. And in an NSF grant that we've been doing for a few years now, we're looking at how number is represented in the brain. In particular, how the knowledge of quantity and the visual depictions of numbers might be confusable or generalizable in different areas. And as you can see in this picture here, we are admiring a large book. So I mentioned how early visual cortex, right, seems to be holding this size information. Well, we want to know a bit more, right? How is this happening? What other areas might be communicating with it? And so to do this, we looked to a technique I developed a while back called informational connectivity. Now, again, I don't have time to delve into all the details of this technique, but if you're familiar with functional connectivity, it's analogous to that. So functional connectivity looks at areas and identified areas that fluctuate together in their neural activity or from the bold response. Informational connectivity does it similar, but from a more of an, as the name suggests, an information perspective. Instead of seeing fluctuating overall signals, it looks at fluctuations in the information over time. So as a reminder back to that original diagram, instead of looking at how there's an overall change in voxel activation, it will look at, for example, the confidence of a classifier or the similarity of a pattern. And importantly, we'll look at this in two different regions and look at it as if they fluctuate together. And actually, if you're interested in this method more, I discussed this with Stefano Anzalotti in a Trends in Cognitive Sciences paper. And actually developing a method, it's kind of, it feels a little, what I imagine it must be like to kind of have a kid and send them out into the world, send them off to school, you pack their little lunchbox, you hope that they make friends and they don't get into any fights. And I'm happy to say that the method, there are other applications beyond what I've used. Other groups have used it to find some really interesting things in the world of memory and perception. And And recently as well, not only is the offspring gone into the world, but it's even had its own offspring. This method's been used to kind of develop new methods, so they grow up so fast. Anyway, I get the point of this. But in my lab, we're continuing to develop new methods to combine multivariate information with connectivity. For example, a new method, representational connectivity analysis. Okay, coming back to size. So why did I mention this? Well, we found significant informational connectivity between early visual cortex and the angular gyrus. We looked at some key hubs here, temporal lobe, or temporal pole, anterior temporal lobe, I mean, and angular gyrus, because these have been proposed as regions that are supramodal hubs, semantic memory hubs. And the angular gyrus shared information, or, shared fluctuations in information after learning with early visual cortex. And this is actually really interesting, because not only is the angular gyrus been proposed as a supramodal hub, but it's perfectly placed to be involved in spatial processing related to the parietal cortex, something that you really will want and need as your processing size. So to summarize these studies so far, have shown that activity patterns in early visual cortex include information about real-world size, even independent of category and visual features. When you learn real-world size, that changes the activity in early visual cortex. And this might come from the angular gyrus feeding information to early visual cortex. And incidentally, this is consistent with other findings in the literature that early visual cortex seems to represent more than just retinotopic information. Or put another way, it's more than just reflecting what's in front of us in our visual field. For example, studies that have used ambiguous stimuli show changes when people understand or add meaning or determine shape within an item, even though what they're seeing remains unchanged. So there's a few other really interesting findings like that. And I would add this, some of the work I discussed here into the mix, that early visual cortex does more than just reflect the visual field. Another form of perceptual variation is really important for what a concept is. When we think of what a concept like a shoe bill is, the label shoe bill not only describes abstract features, but binds together visual commonalities. Right? We understand that a given species will have different individuals within it, and that one individual could be looked at and observed from different angles in different orientations. Could be flying, could be on the ground, and so on. So there are these requirements that if we have a concept represented in the brain, this neural representation must be great at generalizing across visual depictions. It must be robust across long delays, and it must be organized in some way. It's conceptual. Have some kind of information like the category it's from, like bird. When it comes to multivoxel patterns, we have a couple of measures that have been used to examine memory. One is pattern robustness at encoding. And this is basically just how reliable the pattern is in your brain when you're encountering and learning. The other is pattern reinstatement. And that's how the degree to which a pattern is reinstated from when you learn to when you later retrieve. So the maximum reinstatement would be the pattern just appearing almost intact again. And interestingly, these have been examined, but only previously in the memory literature for just static word and image pairs. So in past work, for example, a word might be given, I don't know, a particular word, chair, and it might be paired with a scene image. And that chair scene reinstatement is then examined and like tested, right, to understand memory. But as I've said, memory and concepts are much more than a static word to image pairing. They cover a lot more. It's also the case that a lot of memory studies really only test memory after 5 minutes or 10 minutes, at most a week. And so we wanted to surpass and improve upon both of these aspects as we understand concepts. And the work I'm going to describe here, this study, is led by Heather Brewitt, and also in collaboration with Gina Calloway and Natasha Tokovitz. So in this study, we had people come in for an fMRI scan as they learnt new pairings between new, in this case, Dutch words and animals, tend to be unfamiliar animals. And they actually, the people came back the next day, immediately after the scan, to retrieve and learn during EEG. And I'm not going to be talking about that here. We are analyzing that data, but I'm going to, for time, just not sort of talk about that. But then about approximately a month later, they came back for a second scan and retrieved all of the information they had before. And that first and the scan and the scan at the end of that approximate month is what I'll be looking at today. So these were native English speakers who had no knowledge of Dutch or German. Those are the words, the Dutch or German words that we'll be using. They weren't necessarily, they weren't at all the words like associated with that animal. We wanted to control it in case that gave certain clues. And they were selected in a number of different ways, but they were all unknown to the people. So people were learning these approximately exactly 60 animal Dutch word pairings. And there were about 25 images for each animal. It was really important to us, as I said, to test how well the patterns generalize across different visual depictions. These were from different categories. And during encoding, people were presented with the item and then the word. In this case, it's fles. Then later, they were asked to recognize or test their recognition by being given the animal and word and having to indicate whether it was a match or not, where some items were mismatches. They were also asked to test it on their ability to recall the word as well. We looked at a number of different regions, and we had primary ROIs, which we were interested in based on pretty clear hypotheses. But we also had secondary ROIs. And these secondary ROIs we looked at based on a systematic review of the literature and then corrected for multiple comparisons. So what did we find? Well, first of all, during the encoding robustness, so how reliable the signal was, despite the visual depictions differing, for, let's say, the fles, right, which here is actually the shoe bill, we found across a whole host of regions significantly reliable robustness. But what we're really interested in here is not just if it's robust, I mean, that's great, and that's even required for some future analyses, but we're interested in whether that's important for memory. Does that predict whether someone will remember an item 1 month later? With this more stringent criteria, we found that the ventrolateral prefrontal cortex, very important for, or believed to be involved in semantic retrieval, played a very good predictor, a solid predictor of whether someone would remember that particular item. As did medial posterior cortex, pararhinal cortex, which we know is really heavily involved in binding together features of different objects and so on. And also, again, early visual cortex comes back as well. When it came to the encoding and retrieval reinstatement, so when someone retrieves it a month later, does the pattern look very similar to what happened when they were learning? We actually were surprised and found that there wasn't much here. We had some reinstatement, including in that semantic retrieval area, the ventrolateral prefrontal cortex, but none of these predicted that future retrieval. Why? Well, as I said, these visual depictions are very different, and during recognition and retrieval or recall, we made sure that they were seeing and retrieving and during the scan brand new images, right? So that there really is no perfect visual overlap. And so we found this generalization, at least for robustness. We were able to predict after long delays, less so for pattern reinstatement. And the patterns that I've been talking about, the information that we were detecting was present for the taxonomic category. It's not just the pattern for a particular item is there, but I'm talking here about conceptual information, whether the bird kind of taxonomic pattern is present as they are learning and retrieving these items. So it's really a conceptual property. We're building on and continuing some of this work going forward now. I've been awarded a grant from NSF to look at how this kind of reactivation, neural reactivation, plays a role in whether something's remembered. So it turns out that people are pretty terrible at predicting whether they will remember something, whether an image, at least, it will be remembered. We're really poor at it. Yet, the memorability or the probability an image will be remembered is remarkably consistent across people. If I'm likely to remember something, you are also likely to remember it. And why this is, there have been some mixed findings on. Some studies have shown or suggested that visually distinct items are more memorable, like low-level differences. Others have suggested that high-level content is important, high-level visual content. But this has been really hard to pin down, partly because some of these studies do tend to sort of scramble the high-level content, show a disruption, but it's still hard to pin down then what that high-level content exactly is. So in this next study, to get at this, what we do is we combine a convolutional neural network with cognitive experiments. So a convolutional or neural network, or CNN, you may have encountered some of these in the media or in other work. It's called deep learning. And these deep networks are trained on millions of images. And for the example I'm talking about, they're trained to recognize images. So they'll look, you'll give it an image, and through processing that I'll talk about in a second, it may give a label like field, street, building, so on. And it turns out that the organization of these CNNs shares some similarities with the visual system. They're hierarchical. They tend to extract these features, low-level features at the beginning, which then build up onto high-level features before a given object is categorized. And we're going to be using this because once you have this trained CNN and you've put an image through, you have information at each of these layers, each of these levels. So how we did this is we took 1,000 scenes, images, and we submitted this to a well-known CNN network trained for object recognition. And so now we've got these 1,000 images with information that we can extract from this network about these images. We had 100 participants come into the lab, and these participants were shown images that were either similar or discriminable from others based on the layer of the CNN. You can think of this as like, or it is, prospective assignment. We are letting the CNN decide which images a participant sees. If they're randomly assigned to the layer 1 group, the layer 1 of the CNN will decide or will be used to select the images they see. And similarly for layers 3, 5, and 8, we kind of spread out across the CNN. So participants, depending on what group they were in, saw these images and made simple indoor-outdoor judgments. They weren't told it was a memory test. And after a distractor, they then did a free recall and a recognition test to see if they remembered having seen these before. Oh, and this was led by graduate student Griffin with assistance from Assam. So what did we find? Well, memory for people was significantly predicted, and there was an interaction between the layer and whether the image was discriminable or similar. And with most interactions, it's easier to see it. And what we see is that discriminability in layer 1, which reflects low-level features, and similarity at layer 8, which reflects high-level features, both predicted the memorability. So you're more likely to remember an image if it's discriminable in layer 1 to other images, or more similar in this later layer. I won't go into false alarms, but these false alarm rates kind of went in the direction you might expect. For example, more false alarms for images that were similar in layer 1. We went on to replicate this main finding, by the way. We made a couple of changes. We took out the free recall test to make sure it wasn't interfering, and just looked at layer 1 and 8 because these was from the main findings, and actually found even stronger results in a second experiment that was a replication. So although in prior work, these discriminable features have been associated with memorability, and some have found similarity in other domains, we find that both are relevant. It just depends on what visual level you ask. And so visual properties at different levels are determining whether items will come into our memory systems. So just like before, it was about semantic levels, right, and granularities, and how they are important. This reflects these different levels of the visual hierarchy. So in this next part of my talk, I want to discuss some work that is under the theme of, once we have new knowledge, once we've learned these things, these concepts or whatever they might be, how does that then affect new learning? And I'm going to talk through 3 behavioral experiments that speak to different aspects of this. I'm going to be able to touch on these briefly, but I hope they give a flavor for different ways that new learning is affected. First, I'll be looking at what happens when you have existing knowledge present during learning. Then I'll be talking about the effect of extensive knowledge or expertise. And finally, when you've got semantic relationships between items, how does that affect new learning? So in prior work that I won't talk about much here, I've looked at a phenomenon known as fast mapping, where if you present a new item to be learned in the context of an existing item, and you have to infer the name, you show signs of integrating that new word into memory. And by signs, I mean behavioral signatures. So for example, you might see this creature here, and unless you've been to some of my talks before, in which case you will already know at least what the fake word is, you might not know the real word, but you'll see this and you'll say, okay, are the antennae of the traitor pointing up? You'll see this other creature and you say, well, it's either a cricket or a grasshopper, but so I guess that must be the Terato, okay, or Terato, I guess, depending on where you're from. So that's the Terato or Terator. And so you've inferred that. And you needed, or at least, well, you didn't necessarily need, but you could use this other item to do that. And in past work, I've observed that once you learn this word, very rapidly, you show a slowdown in your response to a related word like tomato or tomato. And that's very quick slowdown, lexical competition, is believed, it's a debated area, but generally believed to reflect integration of the new word into your memory. And incidentally, that doesn't happen if you just have instruction, remember the Terato with the insect, and it doesn't happen if you remove here the grasshopper. If you just have the Terato and are the antennae of the Terato pointing up, you don't see this. You need the second item. And that motivates the study I do want to talk about here. So we looked at the factors that might influence this to really try and understand what is it about this known item. In particular, and this known item I'll call from now on the supporting foil, right? Because it's not the answer to the question, or it's not relevant, but it supports it in more ways than one. And so in this study, one important property in semantic memory is how typical something is for its category. So I presented supporting foils that were typical, like here a fly, kind of a typical fly you might see for an insect, and also some that were atypical, right, like the slug. In this case, you have to read the question and you have to look at both of these to answer. For example, that second question refers to whether the head is above the ground, right? So you might want to process these. But ultimately, the items and the words are still the same across both. And this is all counterbalanced across participants. We also looked at individual differences in spontaneous semantic memory use. So there's a validated survey called the Survey of Autobiographical Memory, which is a self-report survey which measures or has people report how often they use different forms of memory. For example, here are a couple of questions. I can learn and repeat facts easily, even if I don't remember when I learned them, or after I've read a novel or a newspaper. I guess that should be a website now. I forget the facts after a few days. Okay, so obviously reverse coded and so on, but these are items that go into a score for how spontaneously you report using semantic memory and episodic memory and so on. But I'll be focusing on semantic memory here. And I noticed in some work that when we're looking at this, that the brain areas that are associated, the resting state networks associated with individual differences in the semantic memory score look very similar to some of the brain networks that you see during fast mapping. So that kind of intrigued me. And so I looked at both these factors. So let's look at some results here from looking at, I think about 25 people also doing learning and being exposed to fast learning these items with a typical or atypical, while also looking at their semantic memory scores. Well, we're looking here at lexical competition. As a reminder, that's how much your knowledge or how much your response time slows to a known word once you learn Terato, in this case, in the example case. Well, first of all, we saw an effect where you see lexical competition, but only for the atypical items. And again, remember, this is not learning the atypical items. You're still learning the funny red insect. It's all about the supporting item being atypical, but you don't see it for typical. In fact, you see the reverse. But I'm hiding something, which is that when you break people up into groups based on their semantic memory scores, you only see this in those with high self-report semantic memory. We went on to replicate this by going back to one of those earlier data sets, fast mapping data sets, where I didn't really think about typicality, certainly didn't separate it into typical or atypical, but as tends to happen, had a continuum of typicality, and we found that that predicted the lexical competition effect. So these two factors do seem to be really important. Next, we looked at extensive knowledge or expertise. These things, knowing that what a grasshopper is fairly simple, fairly kind of maybe somewhat shallow information. So we wanted to look at what happens or the effect of more extensive knowledge. So prior work on this, and there has been much, but in cognitive science, a lot of this is focused on learning just the target of expertise, like chess pieces on a board for a chess grandmaster. Or when it comes to semantic information, very much explicit learning. People being told, you should now try to remember this. But just like fast mapping, it tends to be what we call incidental encoding, or encoding without deliberately trying to encode. We wanted to see how expertise operated when people weren't told to try to remember different associations. We were also interested in whether, in this particular instance, you can form associations between expertise verbal content and fairly arbitrary, neutral, if you will, visual content. And this is led by Heather. So how did we do this? Well, we drew on sports expertise. Something that's very, readily available. That said, we didn't want to restrict ourselves to people with particular pockets of location knowledge, which will become clear when I show you the design. So we recruited online through Amazon Mechanical Turk. After exclusions, we took the top third and the bottom third of people who scored well in the knowledge test. And this knowledge test was about, sports knowledge test was about things like, the certain positions in the teams, the roles, and so on. Rules of the game, and so on. If I say sports expertise, by the way, I really mean sports knowledge expertise, just to be clear. So people, the participants, saw faces, and these are faces from a face database. They're not related to particular sports in any way. They're counterbalanced and randomly assigned. And each face was presented twice. And there are 20 athletes and 20 non-athletes, right? So here we've got an Indiana firefighter and a Panthers running back. And they're asked pretty simple visual questions. Does the firefighter have short hair? Does the running back have light hair? Visual feature kind of responses. And as you'd expect from something this easy, everyone, the sports knowledge experts and the non-sports experts did about as well as each other. It's worth pointing out here that really we're all, or a lot of us anyway, are experts at the non-sports category here. You know what the role of a firefighter is. You probably know where to go to talk to a firefighter, right? Same for a waiter, and so on and so on. So and it's not that I'm saying we're not experts in this. What I am saying is that the groups varied in whether they were also an expert in sports knowledge. First of all, what we did not see is we didn't see a difference in how familiar people's knowledge of whether they saw a face, just a face on their own. But when it came to associating with that knowledge, we saw differences. So the sports experts were much better at choosing the correct label, the descriptor, like the Panthers running back, okay, from 3 options for a given face. And also during free recall, where they were showed a face and asked, what was the label that was associated with this, the sports experts were much better. And when it came to the non-athletes, like the firefighter, as you could see, both groups did equally well. So clearly expertise is driving not just sort of general familiarity, but very much the associations here. And what I think is particularly interesting, this association was between clearly something verbal content within the expertise, like Panthers running back, and an arbitrary image. It wasn't like a sports logo or something like that would be, clear expertise material, but is selected randomly from a face database. So fairly neutral. So the third of influence of knowledge is influencing semantic relations. So items can be unrelated, like driver, classroom, teacher, racetrack, but they can also be related. They can be in a memory schema. And if we flip these items around, driver and racetrack and teacher and classroom are clearly semantically associated. So in this next study, I'm describing results where we looked at what's the influence of items having a semantic relationship when they're being learned. And I won't have time to go into all the details, but there are several theories that we are comparing here. And in particular, we're looking at when you learn these associations, do you form an association that's symmetrical? So learning it one way helps you in the other direction? Or if you learn it one way, does it just strengthen that and doesn't strengthen as much the other direction? So do we have these symmetrical associations, or are they somewhat independent? And this work is led by Ben Popper. So let's look at the design. Participants had a learning task, and this is really important for understanding the study. It's not just for reasons that will become evident. And so they're learning and being shown different pairings, like teacher and classroom. Then during the they have test cycles until they reach criteria. Now these test cycles showed them, for example, one of these items, like teacher, and they then had to complete, like in this case, classroom. Okay, they complete the response, C, type classroom. Then the next test cycle occurred, and we deliberately flipped the order. Now they see classroom, and they have to type teacher. And this happened very deliberately, alternating for each test cycle. Again, this might seem, you might be wondering why I'm emphasizing. It will be very clear very soon, I promise. And so we then after this, teaching them, we then tested them by looking at their recognition or their ability to tell whether these things were linked. Both things that were studied directly, like teacher classroom, but also things that were linked indirectly. I didn't mention this earlier, but these pairings were not just sort of A to B, teacher classroom, but they were then pairings of 4. So it might be teacher to classroom, classroom to student, and student to auditorium. In that example, a teacher and auditorium are connected kind of indirectly, but they're still linked. First of all, looking at this question of linked versus studied or directly taught, when we look at the relationship or the effect of being related and being in a schema, we found that all the difference happened around these items that were linked. It was forming these sort of distant links that really benefited from items having a relationship or being within the same memory schema. We see the same for response times. So what about that question about how the character and how these associations are formed? Well, if you look at the response times and the accuracy, as participants progressed through that sequence of alternating teacher type classroom, classroom type teacher, and you look at their performance for related in the black dots or unrelated in the triangles, hollow triangles, you'll see quite a different pattern. While the unrelated is smooth, suggesting that as they learned in one direction, the other also benefited equally. In contrast, the related items show this zigzag, suggesting that when you learn in one direction, that effect doesn't transfer to the other direction. So it seems there's a difference in whether the associations you learned are learned symmetrically when it's unrelated or asymmetrically when it's related. In other words, you learn classroom to teacher doesn't mean as well you'll get teacher to classroom. Actually, and after this, we replicated this. We pre-registered our hypothesis, ran another group of subjects, and again replicated this result. I don't have time to go into the details, but we actually have a study that we're writing up now, it's to be submitted soon, looking at the neural basis for some of this. And in particular, we use some of these classifier multivariate techniques to try and find regions that, especially interested in this studied versus linked distinction. And to give a kind of a headline, we find a lot of action happening in the medial prefrontal cortex, a region that's really been linked heavily with memory schema. And this is run or led by John Paulus and also Carlo Vignali, who looked at reward and how this relates to it. So these different ways that existing knowledge affects learning have included semantic properties of another item during encoding, expertise, helping people associate verbal and visual material, and finally, semantic relations between items affecting the character of associations. The final thing I want to talk to you today is about naturalistic dynamic episodes. So naturalistic dynamic episodes, basically things we have in real life, right, are spatial and they're temporal. Unlike an image, well, an image might have some spatial components, but it usually doesn't have temporal components. But these are really important to memory. In fact, if you have some piece of information that's learned in the context of a video, a dynamic episode, you're more likely to remember that same piece of information than learned in a static context. So some dimension, something about it, these dynamic episodes have an influence on memory. So in one study, behavioral study, we looked at how classic influences on memory, ones that I've talked about today from my own studies and also past studies, for example, prior familiarity, just saw that in the expertise study, the effect of post-encoding sleep, so sleeping between encoding and retrieval, in a study I haven't talked about today, but I've done previously, and memory traits, these like semantic memory self-report traits. So we had 115 people come into the lab and they watched silent nature videos and so on. So these are unfamiliar animals and they're seeing these videos. And during the task, they're moving the slider to indicate their interest, just to keep as a kind of a cover task. So after this, people are retrieving what they remember from the videos. They're retrieving immediately after, and they come back after sleep or after wakefulness. We didn't actually deprive people of sleep, had a 12-hour, 24-hour design where we tested them before or after their normal night's sleep. And we assessed memory recall in different ways. Free recall, asking them what they remember, just to give us what they remember. Cued recall, responding to a prompt, or recombined recall. I won't talk about recombined recall today, but for us, that was having people describe like a day in the life of an animal, having to piece together bits from the video in a new way. Oh, and incidentally, these snapshots from the these videos were put together in a really aesthetic way by John Paulus, who's on this paper, and it clearly, the people learning a memory really liked it, and so it made the cover image as well, forming the shoe bill here from different frames of the video. Perhaps I would argue reflecting that the conceptual knowledge, the familiarity of the shoe bill structures our learning of naturalistic episodes, which is the result I'm showing here. Turns out that prior familiarity of animals, how familiar you were with the items in the video, predicted cued recall, but not free recall. And it predicted cued recall of spatial and temporal features, or spatial trending, but especially temporal features of the video. What do I mean by that? Well, temporal features might be what happened after the bird flew. Spatial features might be where was the bird, when this happened, or something like that. Where was the bird here? Or what was the bird next to? And you might be surprised that the other one, attributes, was not related to familiarity, but actually these attributes were selected so you couldn't use prior knowledge. Things like eye color or the particular pattern markings on the animal. So it's interesting that temporal dimension and a little bit spatial, those things that are really central to videos, were really strong predictors of familiarity. While that predicted cued recall but not free recall, we saw the opposite for sleep and the memory traits. I won't go into the memory traits for time, but the sleep, if you look at the wake group, these white bars here, you see that without sleep, you see a loss of information that could be retrieved. With sleep, there's a protection against that loss over time, but only for free recall. We've gone on further to look at what might be happening here. In particular, testing hypothesis of sleep might be shifting the organization, the conceptual semantic organization of material from episodes. And just to briefly tell you a bit about this, we've been analyzing some of those free recall passages using tools like latent semantic analysis. And in particular, we've been looking at how these sentences at the sentence level in these passages are semantically related to each other, both sequentially and just generally. This is work by Caroline Wren in my lab. And to give a snapshot of some of these results, we find that the semantic coherence, or how related items are, drops significantly from sleep, Why might this be? Well, we think that sleep may particularly benefit a free recall through neuronal replay that we know happens after we learn. And newly encoded memories might be reorganized as part of this. And this process of reorganization might allow people to access different features of episodes across semantic space. The very, very final study I'm going to talk about here at the end is looking at neural correlates of some of this. So the neural signatures of successfully encoding naturalistic episodes. Because even after experiencing the same episode, some people will recall certain details while others cannot. So, for example, there might be a particular time period during this talk where some people watching will be able to describe something that happened, and for other people, they can't. So what's happening here? What's reflected in the brain's organization of episodes that makes this change or makes this difference? To look at this, we all will look at intersubject pattern similarity. What this means is we're going to look at the patterns in one individual's brain, and we're going to look at the similarity to other people's patterns. And we're going to be looking for common neural signatures or fingerprints that reflect successful encoding. So we're going to look for what a particular pattern looks like in someone's brain when they successfully learn an episode versus when they don't. And what's really useful for this purpose, the episodes that we gave, just like the example I just had, were remembered by some people and not others. So for every episode, we've got examples of people who saw that episode and remembered it, and other people who saw that same episode and did not remember it. And this is work run by Griff. So in this study, people are watching these kind of videos we've seen before in the scanner. They're encoding as we're collecting fMRI data. They then came out of the scanner and retrieved and were tested on that information. And also they came back the next day and were tested in a delayed way. These were 20 participants in this particular study. So to cut to the chase, we found that people who successfully later remembered an episode, those people had greater intersubject neural similarity with others who also remembered the episode within the declarative memory network. And in particular, if you look at the pattern similarity between individuals for retrieved compared to not retrieved episode, that again, they're watching the episode in all cases, but this is all about whether they later retrieve it successfully. And then once we know that, we go back to that encoding data and kind of code it as, oh, yep, they retrieved this part of the video, this segment of the video. And we did that using cued recall questions. So it might be what happened after the bird flew, and they might give an answer. They might say, oh, it laid an egg. And then we could go back to the video and what's happening during encoding to get the brain pattern during that time. So we see these particular regions, but what I was particularly surprised by was that two of these regions actually contained the posterior medial cortex and the default mode network actually contain episode-specific information. So this is information, not just a general encoding success signal. I can talk about in the questions maybe how we know that, but also why that also it has information that's specific to each episode, and that's across people. Hippocampal patterns were more similar when you defined memory success the next day. Remember, we had people come back than immediately after encoding, and that's really useful and interesting. It supports the role of the hippocampus in the durability of memory, and also shows the importance of this length of delay, this consolidation in this common neural similarity pattern. And we didn't know this before this. People have studied the hippocampus and memory regions within individuals, but we didn't know if that particular pattern was kind of idiosyncratic or was generalized across people for an episode. We show here it's generalizable enough that we can actually see pattern similarity, and actually in a separate analysis classify based on other people's patterns whether someone will remember or not remember a particular episode. And now we're going on after this. We've collected some pilot data in older adults, and we've got some really intriguing findings where it's early days, but it looks like we might, in older adults, might have a signature for encoding failure. In younger adults, we just really had one for encoding success. Older adults might have encoding success and encoding failure, suggesting a common basis for why they might fail to encode the episodes. So that's pretty exciting. Also, this general idea of identifying neural signatures or fingerprints. Here it was for episodic memory or memory encoding success. That general idea of getting a neural signature has motivated an R01 grant with Michael Sayat that very recently got funded, which is going to use these, where I'm going to use these with Michael, these same kinds of techniques to look at neural fingerprints to understand why smokers get craving relief. Some smokers get craving relief when they smell an odor. So we'll be doing scans on a large number of individuals who are dependent on nicotine and then smell an odor, looking at their change in craving and just trying to understand what neural fingerprints are present, especially in those people who get success at having a reduction in craving. So I covered a lot today, but to summarize, I've covered how memory influences a visual system. I did that through real-world size. How visual variation, whether it be semantic levels, high levels, or it could be visual variation through a CNN, influences memory. The different ways that once you have memory, new learning is affected. And how information in busy, bustling, dynamic visual episodes, how that information becomes encoded. So you can't do work like this without having a lot of people who put a lot of work in, and I want to thank those people. I'm sure there are other people who I have missed when I was bringing up this list, and if I have, I am very sorry, but please know that I really do. Thank you from the bottom of my heart for helping with this. I also want to thank my mentoring committee and my tenure committee, Kirk Erickson, Pete Gideris, Chris Shun, and Natasha Tukovitz. I also want to thank my colleagues and the communities and just general kind of intellectual homes of the Department of Psychology and the LRDC. Just having such great colleagues and such a great community is also, honestly, kind of helps keep research fun. And I want to thank the centers I'm associated with, the Brain Institute, CNBC, and the Bridge Center. Not only have they provided community, but they funded some of the work. For example, a Bridge pilot grant funded the fMRI study on schema that I mentioned recently. And finally, I want to thank my external funders, NSF and NIH.